Nice to be back again with you guys. This meeting we are going to discuss enzymes. And the topic enzyme is not only limited to our subject or course Biochem, but also will be discussed again in other subjects like microbiology and other biology related subjects. Although there are some instances that enzymes are claimed as non-living organisms, there are also some point which claims that enzymes are also living organisms. However, we're going to discuss what is an enzyme, the action of enzymes, and of course the factors affecting this action, and also likewise how to limit, how to stop their reactions. Okay, these are the common things that come into your mind if you hear the word enzyme. I am sure enzyme is not new to you. Enzyme was taken already as a topic when you were in high school, especially in your biology and chemistry. So enzyme is a catalyst for biological reaction, meaning to say they either speed up or they retard a certain reaction. But most often than not, enzymes are used to speed up a reaction and then lower the activation of energy. So meaning to say that uh, enzyme do not only speed up reaction but retards the reaction as I have made mention uh, last time or a while. And this property of enzyme is actually capable because of their nature that these are actually protein-like structure. So they could easily adapt, they could easily react with reactions. So in the process of denaturation, simple proteins might be affected. And since enzymes are protein-like structure, they could easily deal with the reactions of protein. Enzymes may contain cofactors such as metal or organic substances, aside from the protein. Now, how are enzymes being named? Typically, enzymes are known to end with the word or suffix "ase" or "ase". So most enzymes actually end with "ase", like for the case of sucrase, lipase, and how do we know, how are we going to identify as two with substance they are reacting with is actually the prefix. The prefix "soc" refers to sucrose. So since ASE is for enzyme, we could tell that sucrase is an enzyme that is responsible in the degradation of sucrose. So you have their sucrase. And then for the case of lipids, you have lipase. There are also some instances whereby the enzymes are not only known through the reaction, but also likewise to their function. So enzymes that catalyzes oxidation are oxidase. Those that catalyzes hydrolysis. So common names of digestion in enzymatic reaction use "in" as a suffix rather than "is". So you have pepsin, trypsin. All of these enzymes are found in the digestive system. Okay, these are some other classifications of enzymes according to their reaction. So you have the oxidoreductases, or it could be oxidoreductases. So they are responsible for the reaction of redox or oxidation reduction. And then you have also transferases. Of course, the reaction is to transfer a group of atoms. And then hydrolysis or hydrolases. And then for the removal of some atoms, you have the lyases. And for the rearrangement of atoms, you have isomerases. And in the combination of molecules, Using ATP, you have the ligases. And then we have also examples of classification of enzymes. You have the oxidoreductases. Example of these are the oxidases and reductases. If it involves both reduction and oxidation, they could be classified as oxidoreductases. But there are those that are only limited to oxidation, and they are called the oxidases. For the specifically for reduction, they have the reductases. And then you have also transferases. The transaminases, transfer amine aces enzyme. So these are the enzymes are responsible in the transfer of amino acid. And then kinase or the transfer of kinetic energy. Phosphate is the one responsible in the production of energy for protein. So kinase is responsible for the transfer of phosphate groups and especially in protein. And then you have also hydrolases. The hydrolases are responsible in the process of hydrolysis. So in the hydrolysis of peptides, you have proteases. Peptides are actually the bonds that connect amino acid from one another in order to form a protein structure. And then you have also lipases. They hydrolyzes lipid ester bonds. And then you have the enzyme lyases, namely the carboxylases and hydrolases. Carboxylases is involved in the addition of carbon dioxide. As for the case of the addition of water, you have the hydrolases. Okay, let's pause. Let's have a learning check. Learning check enzyme 1. Match the type of reaction with the enzyme. Which is for aminase, which is for isomerase, which is for dehydrogenase, and solute synthetase. Okay, I hope you are having your deep analysis right now. Amine, isomer. Okay. I hope this is how you have answered the problem. Okay, aminase. Amine. So it involves in the production of NH3. Amine. Because the amino substance contains the element nitrogen with hydrogen. And then for the hydrogenases, the hydro. So it is the removal. De is removal. Hydrogen. So removes hydrogen atoms. So the answer for letter number 2 is letter B. And then for isomerase, of course, they are involved in the conversion of fatty acid to from cis fatty acid to trans amino acid. It is actually the formation of isomer. Isomer cis to isomer trans. And then for number 4, you have the synthetase. Of course, synthetase means the formation. So it combines two molecules using ATP. Okay, who got 4? Congratulations. Who got 3, 2, and who did not perform, but they simply tried to observe? You should participate more. This is in preparation of your exam. Okay, let's go to one enzymatic action. I am sure you are familiar with this. You have learned this in high school. Uh, an enzyme binds a substrate in a region called the active site. That is the lock and key model enzymatic action. So the enzyme binds a substrate in a region. So the major characters here are the enzyme, the substrate, and the region. Only certain substrate can fit the active site. So the end goal here is actually to join in order to have a reaction. So it is a must that a certain substrate should fit in the active site. And then another is amino acid R groups in the active site help substrate binds. Enzyme substrates, complex forms, and then substrate reacts to form products, and product is released. We can show this as could be seen in the illustration. You have here the key, and you have here the lock, the door lock, and the key of the door lock. So the main objective here is actually to allow the key to enter to the lock. So if the key, if you're going to look at the structure, if the key here has the same structure as this, or it will fit here, meaning to say there will be a reaction. So in this manner, there is the correct fit and it will react. But looking into this key, just alone, simply looking at this portion, it will only fill up to this portion of the lock, right? And then the other part, the tail part, will no longer have a place in the lock and key reaction. The active site should have a rigid shape in order to identify what is the structure of the key and how this could take place inside the lock. The substrate, or which is the key, should have a matching shape as that of what is prescribed in the lock, as shown here. And then uh, the key, the key that fits the lock of the active site, which is actually this one, is actively working here because it fits entirely the shape of the lock. But in this case, for sure, something is missing. Aside from this, it should have this part in order to fill this part of the lock. So in this manner, if this is the substrate, it has no reaction. Okay, so this is again another illustration of the lock and key. So this is the enzyme, this is the substrate. So if these are joined together, the blue portion of the substrate could totally penetrate or could totally occupy or fit this portion of the enzyme. So you have here the E and S complex. And upon the fitting of this or after the reaction, then new substrate could be produced. It could be phosphate or whatever product that it will produce. Okay, we have also another enzymatic action, which is the induced fit model. Again, the structure is flexible but not rigid. Enzyme and active site adjust shape to bind the substrate. It increases ranges of substrate specifically, and shape changes also to improve catalysis during reaction. Okay, so this is the enzyme. If you're going to look at, it is oval in shape. So if this one, the substrate will react with the enzyme, this part of the enzyme will adjust. So it will induce a part in order to have the rectangular shape of the substrate in order to form the ES complex. But once the new products will be produced, the enzyme will go back to its original position or structure. Okay, learning check, enzyme 2. The active site is the enzyme, a section of the enzyme, the substrate, and another question is, in the inducement model, the shape of the enzyme when substrate binds, 1, stays the same, or adapt to the shape of the substrate. Well, you are very right. The active site is a section of the enzyme, and of course, the inducement adapts to the shape of the substrate. Very good. Okay, what are the factors affecting enzyme action? First, you have the temperature. Little activity at low temperatures. That is enzyme. So the colder is the temperature, the lesser is the activity of the enzymes. Rate increases with temperature. So the moment you increase the temperature, the enzymatic reaction also speed up. They are most active at optimum temperatures, that is the human temperature, 37 degrees centigrade. Activity lost with denaturation at high temperature. So it is a little active at low temperature, but very active at 37 degrees centigrade, but it will lose its activity at high temperature. So that is the graph. Both at extremely low and extremely high temperature, the activity of the enzyme is very low. And then another is substrate concentration. Increasing substrate concentration increases the rate of reaction. However, maximum activity could be reached when all the enzyme combines with substrate. So, meron siyang hangganan. So, this is it. So, the moment you increase the number of the concentration of the substrate, the reaction also speed up until it will reach the maximum activity or wherein all of the enzymes have been already reactivated. Another factor is pH or acidity. The maximum activity at optimum pH. Our group of amino acids have proper charge, tertiary structure of enzyme is correct, narrow range of activity. Most lost activity at both high and low pH. So more or less, they have the neutral pH as the highest peak. So the extreme low the extreme low pH, just like the temperature, will retard the activity of the enzyme. Okay, learning check. Enzyme 3. Sucrase, the enzyme for the degradation of, you're right, sucrose, has an optimum temperature at 37 degrees centigrade and an optimum pH of 6.2. Determine the rate or the effect of the following on its rate of reaction. No change, increase, decrease. So, is increasing the temperature of the sucrose will lead to no change, increase the reaction, or decrease the reaction? How about changing the pH to 4 from 6.2 to 4? Will it have no effect or no change? Or will it increase or decrease the reaction? How about running the reaction at 70 degrees centigrade, from 37 degrees centigrade to 70 degrees centigrade? Yes, this should be your answer. Okay. Now let's go how to stop the reaction of enzyme or retard the reaction of enzyme. Inhibitors cause a loss of catalytic activity, change the protein structure of an enzyme, may be competitive or non-competitive, and some effects are irreversible. Competitive inhibition. A competitive inhibitor has a structure similar to a substrate, occupies active state, competes with substrate for active site, and most importantly has effect reversed by increasing substrate concentration. Now, what is a non-competitive inhibitor? Does not have a structure like substrate. It's just the opposite of the competitive. Binds to the enzyme but not active site. Change the shape of enzyme. Substrate cannot fit altered active site. No reaction occurs. Effect is not reversed by adding substrate. And in check 2, check 4. Competitive versus non-competitive. Letter A, is this competitive or non-competitive? Increasing substrate reverses inhibition. Letter B, binds to enzyme, not active site. How about letter C? Structure is similar to substrate, competitive or non-competitive. How about letter D? Inhibition is not reversed with substrate. So that should be your answer. Kinds of enzymes. Kinds of enzymes. You have the first group, the hexokinase. It catalyzes glucose to adenosine triphosphate. Sorry. It catalyzes glucose to glucose 6P with the presence of ATP, degrading it to ADP. So the glucose became glucose 6P, the ATP became ADP. That is the reaction. Or as shown, you have here the glucose structure. Review your structure in carbohydrates. This is your glucose. And the presence of some enzyme, hexokinase, by virtue of the presence also of magnesium, will now convert the ATP to ADP. And the conversion of ATP to ADP, or lowering of phosphate, because the phosphate is given to the glucose, it will have a product here, glucose 6P. So one of the phosphate bond was taken by the 6 carbon of glucose. Okay, you have the oxidoreductases, the transferases, the hydrolases as other examples of enzymes according to reactions they catalyze. So you have just simply to follow the same reaction, no? One carbon will take the coenzyme or the other substrate. And then you have also lyases, and then you have also isomerase, and then you have also the ligases. And then our next topic is on citric acid cycle. The citric acid cycle is also known as the Krebs cycle. It is also known as the Enden-Mayer-Howe cycle. Okay, in biochemistry, your concentration here is on what are the elements, what are the compounds that are actually involved in the reaction, directly involved in the reaction, what are the enzymes involved, and what are the conversions that also happen during the reaction. So the first process actually is condensation. As you can see here, water is released in order to form another compound. And then you have also dehydration. Again, water is removed. Another is cis aconitate in the form of hydration. And this is what you call the process of oxidative decarboxylation. The fourth is still oxidative decarboxylation. And then you have also substrate level phosphorylation. And then the sixth is dehydrogenation. And the seventh is hydration. And then the eighth is dehydrogenation. And the process will continue to condensation, etc. Now, what is the input for condensation? Of course, you have here the acetyl coenzyme A. So you have here the acetyl coenzyme A, and then after condensation, citrate is produced. After dehydration, you have actually two byproducts that were produced. You have the cis aconitate and isocitrate. Because in the process of dehydration, there is a short step of hydration. In the short step of hydration, isocitrate was produced. Okay? And then another oxidative decarboxylation also happens. And the product is the succinyl coenzyme A. And then succinate is also produced as another byproduct or a product rather after the conversion of GDP or ADP to ATP. Okay, so from succinyl coenzyme A, succinate was produced, and then this will undergo the process of dehydrogenation, and it will produce fumarate. After a brief dehydration or hydration rather, after dehydrogenation, malate was produced, and then dehydrogenation will follow, forming oxaloacetate. So that is the process and the product. Now, what are the enzymes that were involved? In the first reaction, condensation, citrate synthase as was used rather as the enzyme. In the second process, aconitate was used, and the next player is also isocitrate dehydrogenase. And then also alpha ketoglutarate dehydrogenase complex was used, and then succinyl coenzyme synthetase was produced or used rather, and then another succinylated or succinate dehydrogenase was used. Then the hydrogenation come in, and then fumarase and malate dehydrogenase was used before it will go to the hydrogenation. So the blue ink here refers to the enzyme involved in the citric acid cycle, and you have here also the products that are produced. So in biochem, what is important for you to know in Embden-Meyerhof pathway or citric acid cycle is the input, the process, and the enzyme. You should know them. So you should know this things. You should know the citric acid cycle. Do not be involved or do not attempt to pass biochem without knowing or understanding the citric acid cycle. Okay? Nobody goes out from biochem, I repeat, without knowing the citric acid cycle. This is actually a video presentation. Okay, that is all for today. Please go and learn your enzymes. Uh, we will go again to another interesting topic before we go to your another macromolecule. This is all for today, and thank you.